Good morning, ladies and gentlemen. You are welcome to PCG 212 Practical Pharmacognosy 1. And today we'll be looking at different diagnostic characters. So we'll be looking at Schedule 6 and be examining diagnostic characters that can be found in the leaves, that's exercise 6A. We'll be looking at how to prepare the specimen for microscopy. We'll also examine the specimen for different diagnostic characters, whether it's the upper and lower epidermal cell types, the stomatal type, the trichomes, and the calcium oxalate crystals, if they are present in your sample. At the end of this lab, you're expected to have learned how to clear your leaf specimens for microscopical examination and be able to identify the different diagnostic characters that can be found in leaves. These are the course resources that are available for you to help you understand some of the characters that you'll be seeing under the microscope. So let's start with the preparation of a sample. One thing that you must consider when you're preparing your sample is whether the sample is in whole form or whether it's powdered. Now remember that these diagnostic characters are constant, so whether your leaf is powdered or in the whole form, you'll expect to find them there, and that's why they are called diagnostic characters. They are constant, and we're able to use them to determine whether the sample that we have is adulterated or not. So if it's a whole leaf sample that you have, you want to separate the epidermis from the mesophyll tissue using either peeling method or scraping. By peeling, you peel off the epidermal layer. For some plants, it's very easy to do. For example, in Bryophyllum pinnatum, it's actually easy to peel out the upper epidermis. But for some plants, you will have to scrape. So you scrape using a new blade. What you're actually doing is trying to scrape off the chlorophyll so that you can actually see the underlying cells. So if you are scraping from the adexal, what you'll be seeing is the abaxial surface. But if you're scraping from the abaxial surface, what we'll be seeing is the adaxial surface. Sometimes it's not easy to scrape, it's not easy to peel, so you have to soak in chlorohydrate or hypochlorite. If you're soaking in hypochlorite, you can soak overnight so that the chlorophyll content will be removed. In chlorohydrate, you can also soak overnight, but if it does not clear, you can actually apply a little bit of heat to your chlorohydrate so as to clear out these chlorophyll content. And when you do that, you rinse off the clearing agent using water, after which you put your sample on your slide and allow it to dry a little bit and add a drop of glycerol and observe under the microscope. If you are dealing with a powdered leaf sample, then you will place a very little amount of your powdered leaf sample using your spatula on your slide. Remember that you do not want to use a lot of the powdered sample. Why? If you do that, under the microscope, you will observe clumps, which not allow you to see these characters that we want to see. So it's just a little bit of the powder, you place it on your slide. Now you add your chlorohydrate to clear the chlorophyll because a lot of the plants are green. So what you want to do with this is pass your slide through the blue flame of your Bunsen burner after you put the chlorohydrate on the slide. Be very careful so that your sample does not get burnt. You rinse off using a few drops of water, that is to rinse off the clearing agent, after which you're allowed to dry and you now add your staining reagent, which is fluoglucinol in HCl. To do this, you add about 2 to 3 drops of fluoglucinol and wait on your sample and wait for about 2 to 3 minutes. After which you now add your HCl. Also wait for about 2 minutes. What you should do now is to dampen off your staining reagent add a drop of water to clear it. Allow that to dry again. Then you mount with a drop of glycerol and observe under the microscope. And so under the microscope, you could observe, for example, these kind of cells. These are examples of the upper epidermis and the lower epidermis of some leaf samples. For the upper epidermis, there was not too much of stomata for this particular sample, but for the lower epidermis for this particular sample, you see that you have an abundance of stomata. Now you will see also of importance that we should note is the walls of these epidermal cells. Sometimes they're straight, sometimes they're wavy. So depending on the sample, you want to note the kind of the shape of the epidermal cells that you have. And so you will also observe the different stomatal types in your sample. For the anisocytic stomata, you will see that the stoma is surrounded by 3 or more subsidiary cells. Those are the cells that surround the guard cells or the stoma. And you will find that one of the cells will be smaller than the other ones. You can see in the diagram of the anisocytic stomata. So that gives you can use that to identify that stomatal type is the anisocytic type. For the parasitic type, you'll find out that the stoma is surrounded by 2 subsidiary cells which have their line of axis parallel to the stomatal pore. So you see that the stomatal pore and their line of axis are parallel. That helps you to identify which type of stomata it is. That it is the parasitic stomata. For the diacitic stomata, you have 2 subsidiary cells with their axes at right angles to the stomatal pore. You can notice in that diagram, you can see that the subsidiary cells are at right angle to the stomatal pore. You also have the anomocytic stomata. In the anomocytic stomata or type, the cells surrounding the stoma resemble other epidermal cells. It means that you can actually identify or differentiate the subsidiary cells from the epidermal cells. So that is in the type that you have in the anomocytic stomata. For the actinocytic, you will notice that the subsidiary cells are arranged along the radii of a circle. They are arranged as if they're circular. Okay. You also have the graminous stomatal type, where you have 2 guard cells surrounded by 2 lens-shaped subsidiary cells. Okay, so that's what you will observe, and this is normally found in monocots. You also have the tetracytic stomatal type. Here you have 4 subsidiary cells that are surrounding the stoma or the guard cell. Sometimes you could have it as hexacytic, where you have 6 subsidiary cells that surround the guard cell. And so this is a pictorial representation of what you will find in, for example, in this plant that I've used here, Populus deltoides, where you have the upper and the lower epidermis. And you can see the guard cells, you can see the subsidiary cells, and you can see the other epidermal cells. You can see, for example, in your A, you see the walls of the epidermal cells, they're straight. But when you go to the sample B, you see that you have wavy epidermal cells. So these are things that you want to note in your sample. Also, you may be able to find calcium oxalate crystals in your sample. And these calcium oxalate crystals come in different shapes. For example, if you look at figure 1.10, you will see the different types of calcium oxalate crystals that are observed. You can see in number 1, you see the solitary crystals. that can be found in allium species. You see this, the crystals are occurring like singlets in just in one. Then you have this the sphere raphides, and in number 2, the raphides in number 3, and the raphides also in number 4, and the sphere raphides also in number 5. And you see the sample in number 6, the calcium oxalate crystals occur like sand spots, and those are called crystal sand. So these are some of the calcium oxalate crystals that you may observe. In the middle, you have under the microscope, these are different calcium oxalate crystals that you may be able to observe in your cells that you observe under the microscope. So it's be able to this is to help you to actually identify these characters. That also, remember that your leaf may be glabrous. If your leaf is glabrous, then it means that you don't expect to see trichomes. It means that it's smooth, it does not have trichomes. But you could have pubescent leaves. And so when your leaves are pubescent, what you'll observe is that you have hairs on the leaves. Some of these hairs you might not be able to see with your naked eyes, but some of them you'll be able to see, but definitely you'll be able to see under the microscope. And under the microscope, you might see them as unicellular, you can see them as uniseriate, you can see them as multiseriate, you might also see them as pilate, capitate, and so on and so forth, as the diagrams indicate. You could also see them as non-glandular or glandular. The glandular trichomes always have like heads, round heads at the top where the oils are stored. So you will get to see these different types of diagnostic characters in. So this particular slide gives you under the microscope some of the trichomes that you would observe. You would observe the first diagram. You can see how the trichome is tapering towards the end. In the top diagram at the top towards my right, you would see that it's a glandular. One of them is a glandular trichome because you can see that head, and you can almost see the oil within it. If you go to the left-hand side, to the extreme at the bottom as well, you will see another glandular trichome. And the examples, other examples, you'll be able to now identify whether they're uniseriate or multiseriate or multicellular. So these are things that you will be able to observe. Now, it's of importance for you to note that when you're looking at your leaf sample, there's some information that you want to look out for, check for. You want to check the shape of the subsidiary cells of the epidermal cells. You want to check their size. You want to check the wall structure of the epidermal cells. Are they wavy? Are they straight? You want to check out whether they have calcium oxalate crystals present. You want to also see if you have some fibers. And so some of these fibers, because you have some vascular elements that you will see as part of your leaf sample, sometimes in your powdered sample. You have them in your powdered sample. They come sometimes frequently in the powdered sample. And so if these fibers are lignified, your fluoroglucinol HCl will pick up the red stain, and you will see these fibers as lignified. So also you'll be able to see the stomata, and each plant has its own type of stomata. Sometimes you can have two stomatal types occurring in one plant, but it will give you every plant has its own signature. So these are some of the things that you need to note on dealing with diagnostic characters in your leaves. So now I'm giving you an assignment, and in this assignment, I want you to identify the different diagnostic characters in the different diagrams that are labeled A to G. In A, what is that you observe? In B, what is it that you observe? In C, tell me what you observe, just in one line. In D as well, the arrow shows you what I'm trying to ask you about, in E and in F and in G as well. So you are supposed to identify these diagnostic characters to be able to know how much you have learned today. Thank you very much.